bitte.
Thank you, Enrique. So it's my pleasure to announce the next speaker who is switching to basic science. This is mainly from Oklahoma City. This is his first visit to Budapest, so special welcome for you to be here. We look forward to the presentation.
All right, thank you very much, Nicholas. Good afternoon, everyone. First of all, I want to thank the host, Nicholas and Peter, for inviting me here, my first trip to Budapest, Hungary. And I want to give a big congratulations for what they have accomplished during the last 10 years. 10 years is not a long time, but they have did a great job and accomplished a lot. And apparently, Nicholas was a big part of that. And looking at those two remind me a lot of my Chinese friends, and what I helped them to develop in the last 10 years. And of course, Dr. Saluja has supported the whole pancreas community all over the world. So we're doing original contribution, and Dr. Saluja has championed the whole world as a whole. So with that, I'm very excited to be here and share some of our recent updates on pancreatic cancer research. Let's switch the gear a little bit from the screen and surveillance of pancreatic cancer. We heard a lot of those talks in this section. So let's go switch gears a little bit to basic science, different aspect from what Dr. Saluja has mentioned. So a little bit of overview of our program, cancer program, from the Stephen Cancer Center at the University of Oklahoma. A few years ago, we received the NCI designation almost hand in hand with the University of Miami. And we're a small cancer center. We only have 3 programs, cancer biology, which I'm leading, and cancer therapeutics, and cancer prevention and control, which is a signature program at University of Oklahoma, because we have the largest American Indian population. And in my program, our members are focusing on signaling pathways in cancer, tumor microenvironment, and mechanism of therapy resistance. So after joining University of Oklahoma Health Science Center 7 years ago, we quickly established the pancreatic cancer program. And early this year, we are finalizing a deal for the Center of Excellence for Pancreatic Cancer and other GI malignancies. This Center of Excellence was led by myself and Liz McNally, and our surgery chair, Barry Addo, from University of Colorado, and Courtney Houghton, my close collaborator and friend, who's the GI chief for working on pancreatic cancer and colon cancer as well. And Ji Jing is our new surgical oncology chief. So this program has been receiving enormous funding from NCI, and in combination, we have published over 300 papers all on pancreatic cancer. We're very excited to move this forward. My own research has been focused on pancreatic cancer for the last exactly 20 years. I started my career in 2002, and then our research is focusing on 3 different areas: early detection, which I'm going to focus on today, how we discover the Zinc transporter in pancreatic cancer, and of course we have other research going on, including but not limited to exosomes, circulating tumor cells, cytokines, and such more. And also second is the mechanism, including Zinc-dependent transcription factors, microRNAs, long non-coding RNAs, circular RNAs, as a regulatory mechanism for pancreatic cancer. And also last but not the least, recently we have a strong research interest on cancer cachexia, including muscle wasting and adipose loss. And also we're developing different therapies, as siRNA therapies, small molecule inhibitors, and combination therapy with surgery as part of the component, also losing diet. So following the outstanding talks from Dr. Saluja, I don't have to do any introduction for this audience, because he has basically covered every ground for pancreatic cancer. And we desperately need novel therapeutic targets for this deadly disease. So today I'm going to focus on surface transporter in pancreatic cancer. I'm very glad that after so many years, the surface transporters finally get some attention. Just give you a few examples. Alex Kimmelman from NYU recently published nice papers on amino acid transporters, and many other groups focusing on glucose transporters. And we have been focusing on heavy metal transporters such as zinc in the last 15 years. And also many groups from Europe, especially from Europe, the people working on cobra transporters such as CTR and ATP7A and B. And also noticed one of the talks earlier focused on SLC26A and different transporters. So this is a schematic diagram of human mammalian cell zinc transporter system. It's a very fine balanced system that on the right side labeled with red or purple. And it's a zinc transporter called ZNT. It's effluxing zinc from inside to the outside, and also put excessive zinc into the storage compartment. As a consequence, the intracellular zinc level was reduced. On the right side is a ZIP family. It's uptaking zinc from outside to the inside and releasing zinc from the storage compartment. As a consequence, the zinc level goes up. So those two protein families work together to maintain the intracellular zinc level as a very, very narrow range. Can't have too much, it's toxic. When you have little zinc, it also causes the different physiological problem, for example, growth retardation and cancer. Later on, I will tell you a little bit more. So we all know that zinc is a very important trace element and the catalytic and structure component used by any enzymes. And 75% of human transport factors are zinc dependent. So zinc deficiency in animals often lead to growth retardation, decreased food intake, impaired DNA synthesis, and severe dermatitis, which is lethal in newborn kids. And also zinc is very important in cancer and tumor growth, since it plays a major role in many transport factors, such as MMPs and CRAB, and many EMT transport factors, which are zinc dependent. So the question we ask ourselves are, what is the expression status of ZIP4 in human and mouse pancreatic cancer? We're very lucky. We work with some important protein which has both human and mouse counterpart. And what is the function of the aberrant expressed ZIP4 in human pancreatic cancer progress? Can we target ZIP4 to treat pancreatic cancer? And the last but not the least, what is the underlying mechanism for ZIP4 mediated pancreatic cancer pathogenesis and progress? So about 17 years ago, we have a very close collaboration with Craig Lobstein, who is the former AP president and the leading expert in the field. And we dig out his famous microarray study, and we found there's a group of protein called the zinc transporter, which is highly expressed in tumor tissues compared with normal, compared with chronic pancreatitis. It's not a dramatic increase, but it's very stable, very consistent across different cohorts from MD Anderson, Michigan, from Mayo Clinic, Hopkins, and China population as well. So we have overexpressed ZIP4 in MIA-PACA2 cells, which is relatively mild pancreatic cancer cell line, which has the least endogenous expression level for ZIP4. We found one single gene overexpression caused a significant increase of tumor growth. This is the orthotopic model. Just look at the tumor size. It's a dramatic increase. And when you knock down ZIP4 with shRNA, not just the primary tumor was significantly reduced, it's just the overall health condition of the mouse was significantly increased. If you look at the control mouse, there's a significant jaundice, loss of body weight, which is typical cachexia syndrome in the mouse. But when you block ZIP4, those mouse looks healthy. So more importantly, we found the overall survival of the mouse was holding the shRNA against ZIP4, which is reduced the level of ZIP4 level, is survival was significantly increased. And we use ASPC, one of the most malignant pancreatic cell line, to establish the xenografts. After a couple of weeks, this first mouse started to die. After 4 weeks, 25% of mouse stay alive. But when we block ZIP4 with shRNA, 100% of mouse remain alive. And also, we recently repeated this experiment in the CAPC mouse model with the CAPC mouse cell line. We established the xenografts. It shows the same thing, that the median survival was significantly increased when you knock down the mouse ZIP4. So next question is, can we use ZIP4 as a target for shRNA therapy to treat pre-existing xenografts? So we found in the control group that were treated with scrambled shRNA, the tumor continued to grow. After 5 weeks, majority of the mouse died of tumor metastasis. But when you treat them with shRNA specific targeting ZIP4, the tumor growth was significantly inhibited. When you open them up, there's still a tumor, but they don't metastasize. So we tried different combinations, ssRNA plus surgery and surgery plus chemo and all three plus together. And on top of that, we tried a pilot study using low zinc diet and compare with zinc adequate diet, see if the diet can play a role in terms of survival. So about 10 years ago, we established the surgical model for pancreatic cancer. Basically, you inject the ASPC, tumor cells, human tumor cell into the pancreas, and about 7 to 10 days later, we do the second surgery, basically remove all the whole spleen and the pancreas body and tail, and everything was done under fluorescent microscope, and the cells was labeled with GLP to make sure all the tumor were removed, and of course, all mouse survived the surgery itself. And but if you just compare the sham group with the distal pancreatectomy, the surgery group, there's not much survival benefit. Maybe it's a few days increase. But however, if you look at the overall body weight index, there's a pretty dramatic increase that the surgery group maintained the original body weight while the control group quickly lose body weight. Once the mouse lose more than 25% of the original body weight, those mouse can die within the next couple of days. So we tried the first combination, surgery plus ssRNA. Now you see a big difference. This is the ASPC mouse model that the control group's immediate survival would increase from 15 to 30 days, almost double. And we also use different cell line, BXPC3, which has a mild, lower level in endogenous ZIP4. And we found the same thing. Immediate survival would increase from 57 to 92 days. So this is the first combination. Other than overall survival, we also took the liberty to collect the muscle cells. That's why I want to, we told in the beginning, we want to switch the gear a little bit from tumor growth to cancer cachexia and muscle wasting. So we found we look at the muscle, the skeletal muscle and TA muscle from the mouse, and also we use a different standard technology that used for muscle biology, which has the tyrosine release and muscle fiber strength. And we found we have a wild type ZIP4, higher level ZIP4, those mouse quickly lose body weight. They go through wasting syndrome, muscle wasting, and the muscle fiber was shrink and must reduce. But when you block ZIP4 with ssRNA, the muscle fiber was rescued to normal length and strength. So I don't want to go through all the detailed mechanism and what does pancreatic cancer cells, which has a high ZIP4, what does it secrete? What does it cause the signaling cascade that works on the distal muscle cells and initiate this muscle wasting? Well, just to give a quick update, and we found the first for P33 was involved in estrogen and MHC1, and those are typical muscle wasting markers. And we also went a little bit upstream and looked at how does ZIP4 overexpressing cause those signal cascade. And we found apparently there's a Rep27B, not 27A, was heavily involved in the exome released HPSG-X cytokine. And the solution has developed a beautiful compound targeting this HPSG-X. And I think our data support this hypothesis from different angles that HPSG-X play an important role in muscle wasting and cancer cachexia. From when we started this, a lot of people think it's inflammatory cytokines, the IL-6, those kind of things. And yes, we do find IL-6 was upregulated when it was high. However, when you block IL-6, nothing happened. It's still cachexia. So we have excluded a lot of other chemokine and cytokine, and we found HPSG-70 and 90 are the major driving force for pancreatic cancer-induced muscle wasting. So as a brief summary, we found ZIP4 overexpression. It's an A transmembrane domain in the surface of pancreatic cancer cells, cause the increased uptake of zinc, and cause the zinc activates CRAB. It's not a one-step activation. It's more like a negative, generate positive, because there's multiple enzymatic progress involved in this simple arrow. So it's more than just a one step. And CRAB activation binds to the promoter region of Rep27B, which is a major player for exome release, and cause the increased secretion of HPSG-X, which acts on distal normal muscle cells and cause the initiation of signaling cascade starting from P30 kinase activation and induce the EBP beta activation and increase the level of UBP2 and atropine. Those are typical markers for muscle wasting. So talking about the cachexia has in the last 5 years and maybe a little longer, that has drawn a lot of attention. That I talked with Stress Cherry many times, and he's an expert on clinical management for cancer cachexia. And it's a multiple syndrome characterized by ongoing loss of skeletal muscle with or without loss of adipose, most times with fat loss, and it caused a significant problem in pancreatic cancer patient. In terms of organ, tumor side, pancreatic cancer has the highest percentage of the muscle wasting and cancer cachexia compared with other tumor types. And also cancer cachexia involves multiple organ, muscle for sure, adipose, and brain, central nervous system, and also liver. That's a part of that multiple organ network as well. Of course, the multiple signaling cascade will involve in pancreatic cancer muscle wasting. Just name a few: the TNF-alpha pathway and TWIC pathway, glucocorticoid pathway, and GDL15 pathway, anaerobic pathways. A lot of pathways going on, crosstalks between different cell types. So this is my pancreatic cancer task force led by Dr. Jing Yang, assistant professor, and Elizabeth Heil, who's specializing in exercise and pancreatic cancer rehab, and Crystal Liu and Benjamin and my senior postdoc working on cancer cachexia. So other than pancreatic cancer therapy, muscle wasting cancer cachexia project I mentioned early, we also have other stuff, other research going on. Just give you a few examples that immunotherapy, we recently published a paper in collaboration with the Wei Chen group and using local phototherapy synthesizing with immunotherapy adjuvant. And we also are the first group that developed this tumor vaccine using virus-like particles using targeting mesothelin as a tumor antigen. We also have developed quite a few small molecule inhibitors targeting different pathways in pancreatic cancer oncogenic signaling. As a quick conclusion, that we feel that zinc level needs to be balanced in mammalian cells. When you have a zinc deficiency, cause a lot of problems, growth retardation, and these dermatitis. But when you have a zinc overdose, first of all, you got zinc toxicity, and second, the chance for developing cancer, for example, pancreatic cancer, and maybe GBM, brain tumor, must increase. So I want to thank our funding source from NCI, from American Cancer Society, from our endowment, and also I want to thank all the people who are involved in this project at OUHSC, my basic science team, my clinical team, led by our GI chief, medical oncology, surgeon, and pathologist. And also I want to thank my collaborator from University of Texas Medical School at Houston, and the Anderson, Baylor College of Medicine, where I worked before, and from University of Florida, the College group, the leading group on Zinc transporter. And also I want to thank my friend and collaborator from Mayo Clinic and Johns Hopkins for providing the tissues, tumor tissues and muscle tissues, which become a precious resource for our research. And special thank Peter and Miklos for inviting me here, and for thank Saluja for introducing them to me. And I really appreciate. I learned a lot in the last couple of days, especially pancreatitis and the screening surveillance. So I want to deliver. Dr. Saluja didn't say that. He's been very humble. I want deliver a sincere invitation for all of you to come over to APA meeting this year. We haven't decided exactly the location. This was a picture from last year at the Amy Rock. And this year will be the first week of November, and most likely will be somewhere in Florida, either Miami or close to Miami. So I look forward to see all of you at the end of this year. Thank you so much.
Any questions for you?
Very interesting story. I wonder what is the actual evidence that the ZIP4 overexpression actually induces a rise in the intracellular zinc concentration? I mean, it could be the ZIP4 also associated with other things. And it's not a simple thing to measure zinc concentrations inside cells. So I just wonder what is actually the evidence that this effect is mediated via a change in the cytosolic zinc concentration?
It's a great question. Actually, before we started any study further, we screened all zinc transporters. There are 16 zinc importer and 13 zinc exporter. We screened all of them in 20 plus organs and different pairwise pancreatic cancer versus benign tissue. We found very interesting, ZIP4 is the only zinc transporter which are upregulated in pancreatic cancer. And the majority of them either remain unchanged or slightly decreased. So ZIP4, it looks like it's a highly tissue-specific zinc transporter. And we also look at for intracellular zinc levels with well-controlled system, control versus overexpression, control versus knockdown. We found there's about 50 to 70% of increase in the intracellular zinc level in pancreatic cancer cell line. And also we checked the human tissue as well. Unfortunately, we didn't find any evidence showing that ZIP4 can serve as early diagnostic marker because the ZIP4 level will remain zinc level remain largely unchanged. But ZIP4 protein was significantly upregulated in tumor tissue versus not.
So can you actually measure zinc concentration?
We can measure zinc concentration using what we call ICP mass. It's inducible mass spec. Can check the trace level of zinc concentration in cell lines, in serum samples, in tumor samples.
So how much, let's say, change in zinc concentration do you actually get, let's say, if you compare a cell with a kind of normal expression and then in an overexpression, how much does the zinc level actually change?
50 to 70%.
And is there any change in zinc transporters that transport zinc out of the cell? Because you have, of course, not only ZIPs,
that's
but
the other totally different protein family called the zinc T family. This is the ZIP SLC is 39. That's zinc T SLC is 30. So it's totally different protein families.
Awesome.
Sure.
Just a quick question. Very nice presentation as always been.
Thank you.
So I'm a little bit about zinc level that's Cody was coming up. So what is do we know what's the cytosolic concentration of zinc in normal cells, in tumor cells, and in circulation?
It's a very good question. In picogram, it's very low because it's a heavy metal. It's a heavy metal, and apparently you can't have too much of zinc. Same thing with iron and magnesium, those type of things. However, we found we did a very interesting pilot study that we want to show that is that ZIP4 overexpressing or is increased the zinc that caused the increase of progression. So we get rid of all the zinc using what we call cachexia, like EDTA, to get rid of all the zinc, and we add back dose-dependent zinc, zinc chloride. And we found in a very narrow physiological range. Of course, when you have a higher ZIP4, and those help the tumor cell grow, there's dose-dependent increase. However, once you reach the threshold, that the cell don't need any more zinc, and they quickly kill those cells which have a higher ZIP4. So the zinc does help tumor growth, but you're going to be careful. There's a very, very narrow physiological risk you can play around with. So we checked the 50 pair of human pancreatic cancer sample along with normal control. Unfortunately, we didn't find any correlation or difference in terms of intracellular zinc level from those patients. It's difficult to understand, but other group, especially from people from UK and from France as well, the study ZIP6, ZIP13, which is a big zinc transporter in breast cancer. This is highly tissue specificity. And they also found the same thing. There's no significant statistic difference in terms of intracellular zinc levels in patient versus not. So we're not ready for prime time for with using zinc as the early diagnostic, but apparently zinc transporters provide a novel insights into into the basic mechanism of how does zinc dependent transport factors promote tumor growth and also cancer in different cancer types.
Great, thank you. So we have some presentations.